Hello, my name is Joshua Mugabi. I welcome you to Biology Remedial Lessons. Today we are on Remedial Lesson Part 4, where we are going to talk about microscopes. What is a microscope? A microscope is an instrument used to magnify objects and make them appear bigger. It's an instrument used to magnify objects and makes them appear bigger. Name the types of microscopes. Name the types of microscope. We have the light microscope and the electron microscope. Those are the two types of microscopes. State the purpose of using a light microscope. State the purpose of using a light microscope. 1. It magnifies and reveals the structure details of tiny objects, such as the cell, that cannot be seen by the human eye directly. Magnifies and reveals the structure details of tiny objects such as the cell that cannot be seen by the human eye directly. Draw a labeled sketch of a light microscope. Draw a labeled sketch of a light microscope. Our microscope has got the eyepiece, it has a body, it has got a focusing knob, it has a stand, a barrel, objective lens, a clip, stage, and a light source. Sometimes we refer to it as the mirror. State the functions of the labeled parts. The eyepiece. The eyepiece is used to look through and to magnify the object. The coarse adjustment knob. It is used to raise or lower the body tube and focuses object roughly. It raises or lowers body tube and focuses object roughly. The fine adjustment knob raises or lowers the body tube by small distances to bring image into fine focus. It raises or lowers the body tube by small distances to bring image into fine focus. The objective lens. These bring image into focus and also magnifies object or image. Brings image into focus and also magnifies object stroke image. The stage. The stage is a platform where object or specimen on slide is placed. Is a platform where object or specimen on slide is placed. The mirror or the light source. The mirror refracts light through condenser and directs it to the objective lens. It refracts light through condenser and directs it to the objective lens. Clips. Clips hold glass slide in position. The clips hold glass slides in position. The body tube. The body tube holds eyepiece and revolving nose piece, which has objective lens. The body tube holds eyepiece and revolving nose piece, which has objective lens. The limb or base. This supports the whole instrument. The base supports the whole instrument. The arm. The arm is for holding when carrying instrument. The arm is for holding when carrying the instrument. The revolving nose piece. The revolving nose piece holds objective lens in place, enabling change from one objective lens to another. A revolving nose piece holds the objective lenses in place, and it enables the changing from one objective lens to another. Explain the procedure followed when using a microscope. Explain the procedure followed when using a microscope. Put the microscope on the bench with the stage facing away from you. You put the microscope on the bench with the stage facing away from you. Turn the lower power objective to click in line with the eyepiece. You turn the lower power objective to click in line with the eyepiece. Ensure that the diaphragm or eyelash is fully open. Ensure that the diaphragm or eyelash is fully open. adjust the mirror until the stage is illuminated with enough light. You adjust the mirror until the stage is illuminated with enough light. Place the slide containing specimen on the stage for magnification. Place the slide containing the specimen on the stage for magnification. Draw the image and indicate magnification of the drawing. Draw the image and indicate magnification of the drawing. State the precautions that are necessary when handling a microscope. State the precautions that are necessary when handling a microscope. Always use 2 hands when carrying it. Always use 2 hands when carrying it. Never place a microscope too close to the edge of the bench. Never place a microscope too close to the edge of the bench or table. Do not touch the mirror and the lens with wet or dirty hands. Do not touch the mirror and the lens with wet or dirty hands. Clean dirty lenses using a special lens cleaning cloth. Clean dirty lenses using a special lens cleaning cloth. Clean other parts using soft cloth or tissue paper. Clean other parts using a soft cloth or tissue paper. Lower power objective must click into position before and after use. Low power objective must click into position before and after use. Do not weight any part of the microscope. Do not weight any part of the microscope. Clean and store well after use. Clean and store well after use. What is magnification? What is magnification? The power of making an image larger. The power of making an image larger is what we call magnification. For example, if you look at the sugar molecule, it has been magnified by 0.1. The sugar molecule has been magnified by 1. The antibody have been magnified by 10. The virus by 100. Bacterium by 1,000. Amoeba by 10,000. A full stop by 1,000,000. And an orange by 100 million. That's what we call magnification, the power of making an image larger. Give the formula used to calculate magnification in a light microscope. Give the formula used to calculate magnification in a light microscope. The eyepiece lens magnification is multiplied with the objective lens magnification. The eyepiece lens magnification is multiplied by the objective lens magnification. Give the reasons for each of the following steps when preparing a cross-section of a stem or leaf for examination under the microscope. Give the reasons for each of the following steps when preparing a cross-section of a stem or leaf for examination under the microscope. Cutting very thin section. Cutting very thin sections. This thin section allows light to pass through, making it easy to observe the tissue. Cutting thin sections allows light to pass through, making it easy to observe the tissue. Using a sharp razor blade during the cutting. Using a sharp razor blade during the cutting. When you use a sharp razor blade during the cutting, the sharp blade does not damage, deform, destroy, or distort the surface of the cell or tissue. A sharp blade does not damage, deform, destroy, or distort the surface of cell or tissue. It makes thin sections, and it also makes thin sections. Placing sections in water. Placing sections in water. We place sections in water to maintain turbidity, hence maintain shape of the cell. The purpose of placing section in water is to maintain turbidity, hence shape of cell. It prevents drying of a section. It prevents drying of a section. Staining the sections with iodine before observing. Staining the sections with iodine before observing. This is to make chloroplasts, starch-containing structures, granules, and plastid distinct. The purpose of staining the section with iodine before observing is to make chloroplast, starch containing structures, granules or plastids distinct. List the parts of the cell that can be seen under a light microscope. List the parts of a cell that can be seen under a light microscope. We have the cell membrane, the cytoplasm, cell wall, nucleus, and vacuole. Cell membrane, cytoplasm, cell wall, nucleus, and vacuole. Those are the parts of the cell that can be seen under a light microscope. Hello, thank you very much for participating in the microscope remedial lesson. Let's meet again in remedial lesson 5, where we shall be talking about the cell. Thank you. Please don't forget to press that subscription button.